Hello friends, welcome to Gynae and Ops lecture. In this lecture, we are going to talk about human fertilization. Fertilization is the process of fusion of male and female gamete, that is sperm and ova. Fertilization starts with the collision of the sperm and ova and ends with the formation of zygote. So first we'll see the structure. So as you can see in this picture, that ova is covered with and that is called as corona radiata, and it is called, looks like a crown, so that's why it is called as corona radiata, the granulosa cells of the follicle of first barrier or first protective layer. The second layer is derived from the protein and it is called as zona pellucida. This is the second barrier or protective layer to the ova. Third is the perivitelline space. So actually it is not a space. To call it as a space is a misnomer. Actually, it is extracellular matrix. Extracellular matrix. Extracellular matrix. And after that comes the actual ova. And this is nothing but secondary oocyte having haploid number of cells with X as a sex chromosome, and it is arrested in metaphase of second meiosis. Here you can see one small structure that is, or one small body used at the time of completion of first meiosis, and it is non-functional cell with the similar number of chromosome as the secondary oocyte, that is 23X. So this is the structure of ova. Next, we'll see the structure of the sperm. So this topmost part is head, which is having nucleus inside with chromosome either 23X or 23Y. Above the nucleus, or we can say at the top of the head, there is a cap which is called as acrosome or acrosomal cap. It is derived from the Golgi body and it is having so many digestive enzyme that will help in the dissolution of barrier or protective covering outside the ova. Next to head is the neck part, and below the neck is the middle piece, which is having so many mitochondria, and because of this it produces so much energy, and due to this energy, this tail or flagella will have the motility. So this is the structure of the sperm. Now, as the ova is released in the peritoneal cavity and sperm is deposited in the vagina. So if we see the lifespan of ova, it is around 24 hours. And the lifespan of sperm is more than the ova, and that is between 48 to 72 hours. And fertilization has to occur in the ampulla of the fallopian tube. So within this lifespan, it has to reach to the ampulla, and within this period only, the sperm has to reach to the fallopian tube to fertilize the ova. So first we'll see how the ova and sperm reach to the fallopian tube or ampullary part of the fallopian tube for the process of fertilization. So first we'll see the transport of ova from peritoneal cavity to the ampullary part of the fallopian tube. So before discussing that, we'll see the structure of the fallopian tube. So here you can see that in the fallopian tube, there is muscle fibers, and the muscle fibers are arranged as outer longitudinal and inner circular manner. This is the lateral aspect, and this is the transverse section of the lumen of the fallopian tube. So here you can see inside is the circular fiber and outside is the longitudinal fibers. So because of this arrangement, this fallopian tube is having peristaltic movement. Next thing is, if we see the epithelium of the fallopian tube, so fallopian tube epithelium is ciliated columnar epithelium. Ciliated columnar epithelium. Ciliated, ciliated columnar epithelium. And the cilia are more abundant or high in numbers or condensed near the fimbrial end. And towards the uterine side, they are present in the form of patches or very scarce. Another thing is, they are the columnar cells, they secrete mucus. So now we'll see that OI is released on 14th day of the menstrual cycle. And around this, there is high level of estrogen. And because of this high level estrogen, it increases the peristaltic movement. And because of this increased peristaltic movement, the fimbria will become more active. And because of that, the releasing ova is easily picked up by this fimbria. The second thing is, uh, under the influence of estrogen, there is increased secretion in the fallopian tube. Also, the cilia or ciliary movement within the fallopian tube will generate a fluid current, and because of that, ova can easily move from the fimbrial end to the ampullary part. Third is, this secretion contains certain chemicals, and those chemicals will attract the ova, and this is called as chemotaxis. The fourth is, because of, so this is the first mechanism, this is the second mechanism, this is the third, and now we'll discuss the fourth one. So the fourth mechanism is a sort of suction. So because of this peristaltic movement, as there is a squeezing and dilatation effect, squeezing then dilatation, there will be negative pressure generated in this fallopian tube, and due to that there will be suction effect, and it will suck the ova to the lumen of the fallopian tube. So in this manner, the uh, ova is reached to the ampullary part of the fallopian tube from the peritoneal cavity. So these are the four mechanisms behind that. Next is the transport of a sperm. So as we know that sperm is having its own motility due to the tail. So its tail moment. And this tail moment is increased because of the capacitation process that we'll discuss in the next slide. That how this capacitation will increase the movement of this sperm. So tail moment, which is increased after capacitation. So this is the first mechanism behind its movement or its transport. Second thing is, as you can see here, that there is changes in the mucus under the effect of estrogen. So under the effect of estrogen, the mucus of the cervix become copious and thin in consistency. Also the pH is alkaline. So all these changes in the mucus are helpful for the spermatozoa to move, or will help in the spermatozoa motility, because if this consistency is thick, sperm will not able to cross this cervical mucus. So this thin consistency and copious amount and alkaline pH will help the motility, or will increase the motility of the spermatozoa. The third thing is the uterine contraction or contractility of the uterus will also facilitate the movement of the spermatozoa. So contraction of uterus or myometrium. So this contraction of the uterus due to oxytocin, and this oxytocin is released as a result of or during sexual intercourse from the posterior pituitary. So from the posterior pituitary, oxytocin is released during sexual intercourse, and this oxytocin will cause contraction of the myometrium and ultimately the uterine body or uterus. The second thing is prostaglandin. This prostaglandin is present in the semen, and that is derived from the prostate gland. So this prostaglandin will also cause the contraction of the uterus. So this contraction of the uterus will help uh, or will facilitate the movement of this sperm. So first is tail, second is the changes in the mucus, third is the contraction of the myometrium, and here the fourth one, that is the outer cells or the cells of the corona radiata will attract the sperm through certain chemicals, and we can say the chemotaxis. So these are the four mechanisms responsible for the transport of sperm. One more thing here I would like to discuss, that in the semen, the total number of sperm present is around 200 to 500 millions. And as the acidity of the vagina, as the vaginal pH is acidic, so because of that, those sperms which are less motile will become very less motile in the acidic environment of the vagina, and they will not able to move further, and ultimately uh, some of the non-motile sperm will not move, and only the active sperm or motile sperm will move to the cervix. Once they will reach to the cervix, because of the immunological action, more sperm or the abnormal sperm will get destroyed through the immunological action of the cervical secretion, or means immunoglobulins present in the cervical secretions. So one first scrutiny is here because of the acidic pH. Second scrutiny of the sperm uh, in the cervix, and further in the uterine cavity, some sperms are phagocytosed uh, with the means within the endometrium or within the uterine cavity, and like this the number is getting reduced, and we can say only 200 to 500 of this sperm will reach to the ova. So this million has been deposited, and only in hundreds they will reach to the ova. Next we'll see that what is happening during the process of capacitation. So during capacitation, two things are occur. One is removal of cholesterol and glycoprotein. So this cholesterol and glycoprotein are derived from the seminal vesicle and from prostate gland also. So because of the removal of cholesterol, the membrane or the plasma membrane of this sperm, as the cholesterol is going out, so this will become more fluid, or we can say the fluidity of the membrane is increased. And because of the increased fluidity in the membrane will become destabilized, or we can say there will be destabilization of the membrane. And this is very important. This destabilization of the membrane is very important for the acrosomal reaction to occur. The second thing is, because of the increased permeability, there is increased permeability for calcium ion, and this increased calcium ion will increase in the motility of the sperm. So like this, the motility of the sperm is increased after the process of capacitation, as we have discussed in the previous slide, and as the tail movement is increased due to capacitation. So this is increased because of the increased calcium influx. And this capacitation is controlled by the enzyme known as fertilization, not enzyme, a protein, that is fertilization promoting protein, and this protein is derived from the prostate gland. So this is about the capacitation process. Now, once the capacitated sperm reaches to the ova and attack the ova, what will happen? First, they will start burrowing of this outer cell layer. And during the burrow process, there is one reaction called as acrosomal reaction. So this acrosomal reaction is nothing but the exocytosis of this cap. So through this exocytosis, the enzyme present in this acrosomal cap will come out. So acrosomal reaction is through exocytosis, and because of that, the enzyme which are present in this acrosomal cap will come out. And the main enzyme are hyaluronidase. Second is acrosin, and third is other proteolytic, other proteolytic enzyme. Now, this hyaluronidase will help in the dispersion of this corona radiata cells, and this acrosin will help in the dissolution of this zona pellucida, and other proteolytic enzyme will help in the dissolution of this matrix of, or you can say the perivitelline space. So like this, the sperm will cross all these barriers to reach to the ova or oolemma. So once the first sperm will come in contact with the oolemma, there is increase influx of the calcium ion. And because of the increased influx of the calcium ion, two things will occur. One is completion of completion of second meiosis, which was arrested meiosis. And second thing is cortical reaction. So because of this cortical reaction, or during this cortical reaction, the cortical granules, or we can say the secretory granules, why it is called as cortical granules? Because they are present at the periphery of this oocyte. So that's why they are called as cortical granules. Otherwise, they are secretory granules only. So they releases, because of this enzyme, this zona pellucida will become hard due to the changes in the configuration of the protein which are present in the zona pellucida. So it will become hard, and it will not allow the other sperm which are still trying to penetrate to go inside. So this zona reaction and the cortical reaction together help in preventing the polyspermy, preventing the polyspermy. So what happened actually? Once the sperm come in contact with the oolemma, there is increased influx of calcium ion. This causes two things. One is completion of second meiosis, and second is the cortical reaction and zona reaction. So this cortical and zona reaction prevents the polyspermy, or due to completion of the second meiosis, the definitive or proper OA is formed, and one second polar body is also produced. So previously it was one, now it is two. So formation of definitive OA with haploid number of chromosomes. Now the tail part of this sperm will remain outside, and only the head part will go inside. Once the head of this sperm will go inside, the membrane will get dissolved, and the nucleus will come out, and the nuclear membrane will also get dissolved of both the cells, like both the gametes, ova as well as the sperm, and they will start arranging their chromosomes, like first with first, second with second, third with third, and in this manner the 46 pattern of the cell is maintained, and this 46 chromosome cell is now called as the zygote, and this is the completion of fertilization. So hope you all are able to understand, and thanks for watching.